So hello to everybody. Welcome to our first webinar organized by the EcotoxicoMicYR researcher group. So today we will host Loren BILLET, which just held her defense in December, and which did her PhD between INRAE France in Lyon and Dijon. So just before to let her start, just to let you know that if you are a postdoc or a PhD student, you are really welcome to present your work if you want. And if so, don't hesitate to contact us with the email below, so EcotoxicoMicYR@gmail.com.
So, excellent, sorry. Hello everyone. I'm going during this presentation to talk a lot about antibiotics. These are pharmaceutical substances used massively to cure or prevent bacterial infection in veterinary or human medicine. Since the discovery in the beginning of the 20th century, they highly participated in the increase of the life expectancy measured. But the success story is a little bit more or less compromised by the increase of antibiotic resistance, which is a processes acquired by bacteria to resist and don't be eliminated by antibiotic. According to some predictions, notably the one of WHO, that will lead to the death of 10 million of people per year due to the lack of treatment options. The antibiotic resistance is increased notably by the action of antibiotics themselves, as they will, by selection pressure, selected the antibiotic resistant bacteria, which are less eliminated and will tend to prevail over the sensible one. And when this selection pressure is coupled with horizontal transfer capacity of microorganisms that will participate through the dispersion of antibiotrophy, antibioresistance among the bacterial tree of phylogenetic tree of bacteria. The second aspect of antibiotics is that they are, for some, poorly metabolized by the body prior to the excretion, and they can return in the environment in concentration, reaching after dilution concentration of nanogram or even microgram per kilogram in river or soils. Those are high. This can have an impact of the environmental bacteria present in those environments, and especially all ecosystem functions, as bacteria are involved in the biogeochemical cycle as one of carbon or nitrogen. And most of all, that will increase the antibiotic resistance in this system, because that will, as we saw before, by selection pressure, increase the amount of antibiotic resistance in the community of environment, but also when the concentration are sublethal, that they not cause the death of bacteria, they will increase the stress in those bacteria, and those, by consequence, the mutation rates of the DNA, and so favors the acquisition of new resistances. And moreover, those effluents do not carry only antibiotics, but they also carry the antibiotic resistance and the gene and bacteria. So when we are looking at the site where the effluents are released in the environment, notably here in this study about a Dutch river, we see that in an upstream site and the downstream site, there is an increase of the gene of resistant SUL1, which is used by bacteria to resist to the sulfamethazine class of antibiotic. All of this increase what we call the environmental resistance, which is kind of a library of antibiotic gene in which pathogen notably can come and take resistance when they need. So if we make the whole story, rapidly after the use of antibiotics, we saw high increase of antibiotic resistance. And now there is a new development, I'm sorry, it's antibiotrophy. What is antibiotrophy? It's the ability of some antibiotic-resistant bacteria to use antibiotics, degrade them, and use them as nutrient sources. It's an emerging function, and the first time that it appeared in literature, it was in 2012, so very recent one, and it was about bacteria that was able to degrade sulfamethazine after exposition of a soil during 10 years of this antibiotic to relatively high dose of the antibiotic. Since there was a whole panel of antibiotic-resistant bacteria who are able to degrade, notably chloramphenicol, erythromycin, tetracycline, incomycin, or beta-lactam, and have energy of it. Not only degrade, but have energy. So the case who interests us was sulfamethazine. It's a synthetic veterinary antibiotic who inhibits the dehydroterate synthase by analogy of its substrate, and it blocks, by the way, the metabolism of the folate, which is essential for the DNA and protein synthesis. It's belongs to the top 5 of the antibiotics the most frequently measured in the environment. The resistance to this antibiotic and all the class of sulfonamides is mainly done by the SUL genes, which code for the same protein, dehydroterate synthase, kind of isoprotein, which are insensitive to the antibiotics. Yeah. And so for antibiotrophy, what interests us, it's mostly done by Mycobacterium genera. And bacteria who are able to do that can degrade a lot of other sulfonamides. And they do it since using well-characterized pathway. And I forgot to tell you that we discovered those kind of bacteria all around the world, in Switzerland, in Canada, or even in South Korea. And this is the pathway. So it's in 2 steps. There is first our molecules with degrade in a dead-end product who is more degrade, and this one who is gonna be degrade after the action of side B and side C, and go in the usual metabolism of bacteria and be full metabolized. So this is the names. But we know all of the things about this function, but it's not so much, and we just don't know how it will act on the problem of antibiotic and antibiotic resistance. In one way, it can be used in bioremediation, as it can be used to decrease the pollution by antibiotics, and by the way, reduce the selective pressure they exert on antibiotic resistance. But in another way, it constitutes an health risk, as it's probably a new form of resistance, and it can also give an additional selective advantage to the antibiotic-resistant bacteria who hold it, and notably increase the dispersion in environment contaminated. So, knowing all of that, we studied the agroecosystemic continuum, where 2 strains of Antibiotrophs have been already isolated, and we want to understand the dispersion of this function along this continuum, which starts by the spreading of manure on soil to fertilize, and who go to the water compartment, aquatic compartment, with climatic events. And we were also to highlight eventual interdependencies with antibiotic pollution and antibiotic resistance. We particularly focused on 2 key interfaces. which are hotspots for the dispersion of chemical and biological contaminants. That is to say, the manure spreading on soil and the soil erosion to the aquatic compartment. Those two steps also called for the concept, ecological concept of coalescence, which are very interesting concept, and which defined how the meeting of two ecosystems defined by their own physical and biological parameters meet in a new one, forming a new one, a third one, different by with its own parameters, biological and physical. And our equation, her hypothesis was that the dispersion of the strain will be notably favored by the presence of the antibiotics, as it will act as a selective pressure for antibiotic resistance, and will also we also hypothesized that the coalescence events and the disturbance, physical and biological, will also promote the dispersion of antibiotics. So for our first experiment, I focused on the first step, the manure spreading on soil, and to do that, we selected 4 soils in Bourgogne-Franche-Comté. One of the soils had already received manure 6 months prior our experiments, and we adopt a microcosm approach to control the, as we can, all the parameters. So in this microcosm, we add the Antibiotrophy, thanks the model strain Microbacterium SPC448. I will call her C448 and probably say she because I'm very close to this bacteria. And then we introduce for half of the microcosm the selection pressure by the addition of sulfamethazine at a high concentration, 100 milligrams per kilogram of soils, unrealistic for probably unrealistic. And then we introduce manure for half of them. So it results in 160 microcosms, which were incubated for 1 month prior to make chemical assays to see the degradation of the antibiotic eventually, and also to evaluate it by other biological approach, the impacts of the treatment on the global antibiotic and antibiotic-resistant community. And in parallel, we duplicate half of the microcosm, the one who received all the antibiotics, and using a radiorespirometric approach with sulfamethazine radiolabeled, we measure during the months of incubation the degradation of the antibiotic. So let's start with the results. So what we saw during this experiment is that here is the installation of our strain of interest, C448, that we approach with the quantification of the gene of degradation, said A. And we can see here in 3, sorry, I will present only 3 because 2 are exactly the same and it's like on the slide. So when the strain came alone in soil in blue marked, she failed. She can't establish in soil alone. But when she was accompanied by sulfamethazine, she always succeeded. So the sulfamethazine, the antibiotic, was essential for the establishment of the strain in soils. But, because as I said before, there is always a but, we were very surprised by the mineralization of the antibiotic during the incubation. Here I present the mineralization kinetics. We can see the degradation of the antibiotic along the month, and we saw that without manure, it's the dotted line here, and I can say it's behind the red line, that there is no or very few mineralization of the antibiotic when there is without manure. But with manure, there is always a mineralization, which is observed. And we have also a soil-dependent response. That is to say, there is probably underlying processes involved. So to understand this, we looked at the community response, the whole community, and here are represented the impacted taxa by sulfamethazine and manure. For this first one, you can see the list. It's a list of all the taxa who've been impacted, their abundance diminished when they are red or increased when they are blue. We can see that there is an impact of the antibiotic, which is soil dependent. In soil A, there is 55 OTU which are diminished, only 8 in the soil C and 20 in the soil D. For the other one, here it's the impact of manure, and we can see squares blue, notably in soils A and C, very strong one, indicating with other evidence that I will not show you yet, that it's probably taxa brings by manure which establish in soil, but not in soil D. And just to remember, it was the soil who already received manure 6 months prior to the experiment and where they are already present. So I made a little kinematics to explain better the hypothesis we made about this result. We just saw that our strain alone is not competitive enough to establish in the community, but not even more the case when there is the antibiotics. And it's probably because the antibiotic by biocide impact will reduce the abundance of some taxa, so will release some niches in the bacteria community. The strain can take those taxa, and by the way, it supports its establishment. The strain, notably in soil A and D, where the impact of the antibiotics is stronger, didn't need to mineralize the antibiotic to establish. It was sufficient. But in soil C, there is less, which is less impacted, the niche release were lower, and so the strain, Mycobacterium, need to degrade a little bit the antibiotic as she have to compensate the lack of a niche released. Up. And when we add more complexity to this scenario with the addition of manure and especially bacteria of manure, we can hypothesize that those bacteria just took the niche released by the antibiotic and force Mycobacterium to degrade the antibiotic. And the not remember the word, sorry, but in soil D it's not more the same. There is no bacteria bring in this soil, so the niche remains free, and the antibiotic is very little degraded. So what we saw about with this study is that the antibiotic supports the establishment of our antibiotic strain in the soils of community bacteria for two reasons. First, because by its biocidal action that will reduce the competition in those communities. It's the same for all the antibiotic resistance. It's because the strain is antibiotic resistant that she benefits of this biocidal action. But the strain is not only antibiotic resistant, it's also antibiotic, and it will use, when the first condition is not enough, of the antibiotic as exclusive nutrient source. So the main conclusion is that antibiotic would increase dispersal of potential compared to antibiotic resistance alone. If you want to know more, there is a paper in Frontiers in Microbiology, and I will be glad to respond to your question, of course. But I have to highlight that there is some limits of this study, of course, and notably because we cannot distinguish the abiotic from the biotic impact of manure, and notably probably manure as impacts on the bioavailability of sulfamethazine, or probably have favored co-metabolism. So that's it for this part. Let's start the second one. I probably drink a little, sorry, forward. Okay. So with knowing this limit, we start the second part, which was about the soil erosion to sediments. And we especially interest, it's the same hypothesis that I already present you. We particularly want to make the distinction between the biotic and abiotic impacts during this coalescence event. So we took two sediments, one in the Ain River and one in the Arban River. The first one be coming from an anthropized site, so probably with a microflora used to the antibiotics. And for the Arban, it was in the middle of the Alps, so probably preserved from contamination. For the soil, we chose the soil C in order to keep the idea of the continuum. And to make the distinction between abiotic and abiotic consequence of erosion, we sterilized the soil and for half re-inoculated with this community. So we adopt here again a microcosm approach in which we put we mimicked the erosion by adding the clay fraction of our soil that we obtained by pedological mechanical separation, mixing. And so we add sometimes the antibiotics, or the antibiotics accompanied by the strain. Never the strain alone, as antibiotics seems to be essential to this establishment. And we here again duplicate and to follow the degradation of the antibiotics with microcosm radiola belt. So let's start the results with the surprise we obtained during this work is for the condition without the strain inoculated. We noticed that during the mineralization, we observed here start of mineralization. For those graphs, there is just the erosion is just labeled by tunnel of blue, but don't take care of it. That's no more important. But we can just notice for both sediments that there was a degradation, a start of degradation. And we thought the same here. So that it's more complicated, this one, but it's a chemical assay realized at the end of the months of incubation. You have here 3 bars for the kind of erosion, and here you have the hatch in hatch, it's the ADMP, the dead end product, and full color is the sulfamethazine, and the dotted line is the amount of sulfamethazine added initially. So we can see that most of the sulfamethazine was retrieved under ADMP form for the Ain River, and for the Arban, even if sulfamethazine was the dominant form, there was ADMP that is synonymous with degradation. It's not a natural product. And we had also another confirmation of this observation by re-incubation of our sediments with a new dose of radiolabeled sulfamethazine, and this time it was high rate of mineralization, immediate, and in mainly all the conditions. So our 2 sediments have endogenous capacities of antibiotrophy with sediment and erosion-dependent variation, but we not go into the details. So when we look at the gene SAD-A, which is the only known gene of degradation for the pathway of degradation of our antibiotics, we notice that in condition without treatment, it's not detected, but it's always detected in the other when there is sulfamethazine, and even more when it's modalities where there is degradation, actual degradation. So the degradation is probably supported by the SAD-A gene here again. So we conduct logically isolation, and we done them by cultivate on a selective medium where the antibiotic was the only nutrient source. And we obtain one Antibiotic strain per sediment. And they are all sulfamethazine resistant, but it's probably a limit of the isolation method, as you have to be resistant to be isolated. And what we can say, very interesting, is that they look the same as Mycobacterium SPC448, notably thanks to those 16S rDNA fragment digestion profiles, which are kind of fingerprint for bacteria. And that's striking, but not so much as all the other antibiotics known for the moment are Mycobacterium. But it has to be noticed that it's not our strain. We made some control to notice that it's not our strain, notably because it has a double selection of antibiotics, so we know it's not our strain. But which interested first was the exogenous antibiotrophy, what we bring with our bacteria. So what we saw here again, the degradation, mineralization during the time. So you have just what we saw before here with the solid circle, and now it's with the strain, the squares, and we saw that the mineralization here is systematic. Whatever the erosion modalities, it's more important and it's immediate. You don't have any lag time. Moreover, there is sediment, it's sediment and erosion sediment dependent. Here for the Ain River, you have about 30% of the antibiotic which is mineralized against more 20 in the Arvan, and with a strong impact here of the erosion. For here, it's the same as already present, so don't be scared. And you can see here, it's mostly H color, so it's ADMP, it's a dead end product, so it's an almost complete degradation in the sediment with the strain. And it's performed by our strain because we count her by quantification directly her on plate with the double selection pressure. And so it can establish a more than 6 log in the sediment, which is kind of important. These analyses are still in progress and will be explained and completed notably by the sequencing data, but they are very interesting, and I will not present you so much because of the time limit. I think I'm already dead, but so here I just represented the divergence of bacteria community depending on the different treatments, and not going more in detail, but you can see here the yellow points, non-treated community are very different from those who receive the treatment, and you have also difference among the erosion modalities. So I will not say more. The last point of my thesis is that we use those different experiments to approach the notion of coalescence, which interests me a lot, and notably how it will act on the spread of antibiotic and antibiotic resistance. We want notably to establish, do bacteria actually circulate between compartments? Seems logical, but not so easy to demonstrate. And the sulfamethazine increase the invasion success and antibiotic resistance between the different compartments. So to do so, in the first study, I use the sequencing data here again. Here are listed all the bacteria whose abundance were increased by manure application. And you have green circle there for each soil, which inform on the strength of the impact, more bigger, more increased abundance, increased, and they can be stored in the circle when the Oteus is absent from the soil initially. For instance, the last, on the right, you have the rank of those Oteus in manure. For instance, for the last one, you have the Pseudomonas, which were increased in the fourth soil, which is the most abundant in the manure, and which were absent from the soil A, B, and C, but not the D one. So what is very interesting on this figure is that there are several Oteus for which have a kind of a pattern. That's to say they were increased in many of the soils where they are initially not detected. The red star. They are systematically present in the manure and moreover in high ranking. And they are not impacted in the soil D. Here you can see there is almost no green circle, and they were already detected in it. So all those kind of evidence makers hypothesize that it was actually manure-borne Oteus that established in soil. But there is a limit. There is a limit of detection threshold which can prevent us to make the distinction between bacteria who just benefit from the input of nutrients, for instance, by manure, from the one who were bought by manure, which just appear after manure spreading in our data, but we can distinguish them. Another point about the sequencing data is that some of those manure-borne, supposedly Oteus, were even more abundant with the sulfamethazine, as they succeed more with the presence of the antibiotics. So probably we can hypothesize that they are antibiotic resistant, but here again the limit. Are they really antibiotic resistant? Because it didn't, the sequencing data didn't inform us about real function of the bacteria. So to do our second experiment, we had a more complex experiments to deal with these two limits. Notably, we had a steroid erosion modality in order to deal with the first limits, and a second, for the second limit, we had a selective culture step with before sequencing to quantify and identify bacteria which are resistant and which are not resistant, but only trying to work on cultivatable bacteria. So finally, the conclusion. So what we show during this thesis is there is actual bacterial circulation between the compartment of agroecosystem, and that notably by the survival of manure-borne Oteus after 1 month in soil, and probably more for those who adopt of resilience with the soil D, which possess again bacteria of the manure, and that this circulation may be impacted by sulfamethazine. That's notably the case for our bacteria of interest, C448, as she can circulate between the compartments, but with the help of the antibiotics, supported by its antibiotic resistance alone, also time when it's not enough, reinforced by its antibiotrophy. So what we can, what is questionable is this antibiotrophy function. Is it a backup alternative for the strain? And notably, we can just respond to this question just by making invasion tests with mutants of the strain, which can be just resistant, just antibiotic, both of neither or neither. And if we arrive to do this kind of mutants, it will also help to respond to the famous question: does antibiotrophy is antibiotic resistance? There is also the question raised by our results is how does bacteria modulate its function of antibiotrophy as she is not always using it. So we can make an experiment, for example, by cultivate the strain in different richness of medium and study the transcriptome or proteome of the strain, notably the SAD genes. And we can also imagine something in the reality. Here we work with microcosms, so it's far from what happened really in the environment. And there is, what about the question, the concentration? Because we worked with 100 milligrams per kilograms of soil or sediments, which is higher than what we found in the environment. So is it the same in the, it can be the same in the environment? Does the strain can just spread with this concentration? And it's not finished. Sorry. What we saw during this thesis is that antibiotics have been found all around the world, and now we have French one, so it's good. Coming from two sites who are not connected to each other, and it's already again microbacterium species with the SAD gene. It's kind of weird. It's striking similarity, and that seems that it's not a spontaneous following the exposure of 1 month during our experiments to the sulfamethazine, and it confirms that there is a strong dispersion potential of this function, and probably an underestimation. We didn't look for it 20 years ago, but probably we not know about it. So, but we can just make some hypotheses. Probably it's because of effluent contaminations in the sediments. It's kind of logical, probably, but in the Alps there is cows in the pastoral system, but there is a question why those cows will have in the gut microbiota similar bacteria than the soil of a Canadian experimental site. So kind of strange. There is also the atmospheric dispersion, which has been shown for other bacteria and function. And there is a hypothesis of an older evolutionary history of dispersion sustained by two recent published work, as sulfamethazine is not so much unknown from the life, as it was produced in a kind of produced by sponge, and that microbacterium can make some symbiosis with sponges. So why not? So that's on this mystery that I want to thank you all for your attention, and thank my two supervisors, my two teams, the members of the project Antibiotics, and the EcotoxicoMic for welcoming me for this presentation. Thank you.
Yes, thank you, Loren, for this really nice talk. And if anybody on the which is online want to ask a question, don't hesitate to put your question in the chat, or you can also ask for directly ask a question. Okay. Is there any question in the room already? Yeah.
The genes that code for the degradation of the sulfamethazine are clustered together?
Yeah, they're all near each other. Yeah.
And you don't see, so that would be an ideal situation for gene exchange with other microbes, but you don't see that?
No, when we quantify the strain microbacterium, we quantify her by 3 ways, and we used the sequencing of quantify by PCR of the SAD gene, the GRO-L gene specific of the strain, which is common gene, I don't remember the name, and we quantify also the strain by plating her. and there is no changes in the ratio between all those kinds, so it didn't orientate with to a possible horizontal transfer of the gene. But we didn't notice it.
But if you look from your information database after okay. If you look for bioinformatic database, did you find those genes in other bacteria in order to understand
we looked for it, but we just found microbacterium, other microbacterium, and it was highly preserved, like more than 99% in all the bacteria we found. After it was just 70% of similarity, so no more.
So only microbacterium.
Only microbacterium.
So your hypothesis, maybe the hypothesis is that this gene is not really transferred.
That's the point of the last slide, that we don't know actually, or probably I don't know if it's just the strain who acquired the function recently, as there all those microbacterium are which are antibiotic are phylogenetically closer to each other than to other microbacterium. So probably it's a recent acquisition,
yeah.
And just a spread of the bacteria in the 10th year, since last year.
Recent or nascent acquisition due to the presence of a similar molecule in the sponge?
Yeah, just it was fun to, when I made my bibliography, that I noticed that 10 years ago, the team just discovered that there are analogs of sulfamide who are produced actually by sponge, but it's not considered as a natural antibiotic. It's synthetic one, which were discovered in the 1930s. That's not so. It's not unknown from the life.
If there is no more question in the room, I will just take the box. Okay, so we have question online.
Pascal, you can ask your question, but you have to switch on your microphone. Or not. Ton micro, il est coupé, Pascal, si jamais tu veux poser une question.
I will just ask in between. In your first part, you spoke about simazine. You spoke about SMZ.
Yeah.
And I thought, for example, with triclosan or another biocidal product, do you have any idea about the results it can make too, or for example,
to add to the dispersion of bacteria, you mean,
or with the results you got with this one, if it can be transferable to other products, or if it is very specific for you?
But not with my strain, as I don't think she have it have other antibiotic abilities, but probably with other antibiotic pollutants, or even not antibiotic pollutants, because other xenobiotics as pesticides or other can promote the dispersion of bacteria that degrade the substance, as soil can be very competitive environment, so being able of degrade a xenobiotic is kind of an advantage for those bacteria to develop and spread. So why not?
No more question? Okay. So we will stop the the webinar.